All right. So this chapter is basically about genetics and evolution. And these are the terms that we will need to know. We will go through it one by one as we go, like homologous. We talked about this before, like alleles. We went through it before. Homozygous, heterozygous, we will see it, probability, genetic tools, genetic interaction, whether it is pleiotropy or polygenic or codominance, incomplete, or so on. We will go through all of those and we will understand what does it mean. So this is just an introduction to tell you it's not really one gene responsible for one thing. It doesn't have to be like that. Some inheritance will happen through one gene, sometimes two, three, four, sometimes it's huge amount, like there is for the skin color specifically, there is more than 100 gene that's responsible for that. So this is just a revision of what we talked about before, which is what is the gene? The gene is what provide instructions for human traits. Traits means um, um, like something like the skin color, like the hair color, like the height and everything. All of these are called traits, like characteristics, physical features, and also functions. And we discussed in the previous chapter how the sperm meet the eggs. After we talked about the mitosis and then meiosis, and how it's going to be 23 in the sperm, 23 in egg, and they merge together to make a zygote, which is 46, and so on. We talked about that. So the offspring or the kids are going to have half sets of the genes from the dad and the other half from the mom. And it is basically a segment of DNA in a chromosome. I hope you guys still remember that. It's extremely important to know what is the gene, what's the DNA, what's a chromosome, what's a chromatin. These are different terms that we have to know. So DNA, a segment, the DNA is different segments. Each segment is a gene that have specific instructions for a specific trait, can be for a specific protein. This is a gene. It's part of the DNA. DNA wrapped around protein, we call it chromosome or chromatin, depending on is it in a, regular part of the life cycle, or is it during cell division. So here are the basic terms that you have to know. These are basics, so you need to understand this. What is this? This is a pair of homologous chromosomes, and we talked about that in the previous chapter. Homologous, it's homo. Homo is the opposite of hetero. Homo means similar. Hetero means different. It's a prefix that you have to know. Homo. So this is homologous chromosomes. Like two chromosomes, it's a pair of homologous chromosomes. They have the same shape. Look at this. Like here, you will see like a neck here, and then you will see this is longer than this. And look, even the segments are equal in size. So it is same shape, same length, same set of genes. But it is a very important word or very important part to know. Same set of genes, not identical genes. Meaning, if this yellow gene here is for hair, this gene is also for hair. I didn't specify. I didn't say for yellow or black or brown. I didn't say that. So same set of genes means the genes that are responsible for something. What is that something? It can be different. So you need to distinguish between those. One of those will come from the mom, and the other one will come from the dad, male and female. One from the male, one from the female. One coming from the sperm, and the other one is coming from the egg. So this is a very important definition to know. A pair of homologous chromosomes. What does it mean? Same shape, same length, same set of genes, one from the female and one from the male. Now, there is another word that we have to understand, which is alleles. What's the alleles? We have like in this pair of chromosomes, we have pair of alleles. This like yellow and this yellow. These are alleles. And alleles is the chemical form of the gene. Does it mean gene? Yes, but we are talking about the chemical form, like the actual chemical structure. Uh, is it a chemical structure that's going to have instructions for yellow hair? Is it the allele that's going to have the instructions for red hair or black hair or whatever? So this is called allele. So what's allele? Chemical form of the gene. Does it have to be the same? No, this is coming from the mom, this is coming from the dad. So maybe this is yellow and this is yellow. This allele is a gene, a chemical form of a gene for yellow hair. And this allele is a chemical form of a gene for yellow hair. Maybe this is for black and this is yellow. Maybe it's brown and brown, black and brown, whatever it is. It's different things. It can be different, it can be the same. This is not our issue. But both of them are alleles because both of them code for a specific trait, like hair, like skin, like anything else. The other definition is locus, and locus is single for loci. It's always like that in Greek and Latin. If you find something that end up with us, if you remove it and put I, this will be the plural for the word. So locus is like location. It's even sound the same. It's location, locus, location. Fortunately, it's the Greek is similar to English in this one. Locus, location. It's the location of the specific gene on a specific chromosome. Like, look at this green. This is a location of the gene in this specific chromosome, and it's located in this part. And here is the correspondent one on the other gene. So it's located here. If it is located like here, like the brown, this is another locus for the gene. The other thing is, I was mentioning here that those, let's say, just assume that those two are for the eye color, and this is for black, and this is for blue, or both of them for blue, or one black, one brown, one green, one blue. What are the expectations of this person regarding the eye color? How is it going to be? Well, it depends. Some of these colors are dominant, meaning it can overshadow, it can take over, it can have the upper hand, and the other one have the lower hand. So here is an example. This person here have blue eye, and the blue eye is recessive. So recessive means weak. This is a weak one. So here is, let's say this person, let's look at the genotype here. This has to be blue, code for blue eye, and this also has to code for blue eye. Because look at this here, this is brown and black. This is dominant. So basically, with dominant mean the strong one that have the upper hand and mask the other one. So what if this black and this is blue? This person will have black. How about if this is brown and this is blue? This person will have brown because black and brown in this case is dominant. It's going to dominate the other one, which is recessive. Understand these terms. We're going to build on it, and it is the core of the chapter. So we have to understand it. So dominant and recessive. Okay, homologous chromosomes, locus, pair of alleles, recessive dominant. This is what we covered so far. So you need to understand that. And we will build on it. As usual, you can stop me anytime you want if you don't understand what I'm talking about or you need more clarification about something, that's always okay. So understand dominant and recessive. If both alleles are the exact same thing, this is for blue, this is for blue. We call this is homozygous. This allele is identical to the other allele. This allele in this chromosome is identical to the allele in the other chromosome. Okay. If that's the case, we call it homozygous. Example, just to make more sense. This is for blue, this is for blue, homozygous. This is for black, this is for black, homozygous. Brown, brown, green, green. If both alleles code for the same thing, this is called homozygous. Homo means similar, homozygous. But wait a minute. You said blue is recessive and blue is recessive. Yeah, whether it is recessive or dominant, if both of them are the same or code for the same thing, we call that homozygous, homo. If it is different, we call it hetero, and hetero means different, heterozygous, heterozygous. So homozygous, heterozygous. Both blue, homozygous. Both black, homozygous. One black, one blue, hetero, and so on. So the alleles here are different. One that's coming from the mom, it can be the same if it is the same, like the allele coming from the mom is for blue eye. The allele coming from the dad is for blue eye. Okay, this is homozygous. What if the allele, or I mean the gene, allele, I'm sorry, the allele coming from the mom is for brown, and the allele coming from the dad is for blue. This is heterozygous, and the result is the dominant will take the upper hand. Here it is, the dominant allele masks the recessive allele. So black and blue, black, definitely black. This person will have black eye, for example. And look at this. This person here are the two chromosomes, pair of homologous chromosomes, because they look the same length, same location of different things, and so on. But look at this one specifically. Here is blue, which is this allele is coding for blue eye, and this allele is coding for brown eye. Can we expect what this person is if I have this chromosome and he didn't show me the person? You're just showing me a chromosome and telling me this chromosome is for this person. Can you tell me the color of the eye of this person without seeing him? Yes, I can. If one brown and one blue, brown is dominant, it will overtake, it will mask the blue. So I will tell you definitely in this case that this person has blue eye. I mean brown eye, I'm sorry, brown eye. How about this? The first one was heterozygous. This is homozygous. Brown, brown. It goes without saying. It's brown, of course. This is the only situation where this person will have blue eye if this is homozygous, recessive. So both of them, there is nothing dominant to mask them. So both of them are recessive, and this is homozygous, recessive. Both of them are recessive. So this person is the only condition that will have blue eye. So we can actually tell from the gene what is the actual trait or characteristic. We call what we see here a genotype. You will see it. Like, what's the genotype for this person for the eye color? Here is the genotype. This and this. What you see on a chromosome. What's the genotype of this person? It's right here. This allele and this allele. Brown, brown. How about this? Brown, blue. Okay. The actual eye color that you see on that person, that's called phenotype. Phenotype. Another term here, genotype, phenotype. Like if I ask you, what my eye color is brown. What do you call this? My actual eye color. You call it phenotype. If I give you the chromosome to look at the alleles, what is it? This is the genotype. Both of them are for the same thing. Both of them are for the brown eye. But what you actually have, the characteristic that you actually have, is called phenotype. Pheno, shape. What's actually there? It's already expressed. But if you're studying the genes for that color, it's called genotype. So the different ways it can be, if it is homozygous. Homozygous means both alleles are the same, but both of them are the same, but both of them dominant, like brown, for example. We call that homozygous dominant. Homo similar, homo same. So both of them code for the same thing. Brown, blue, homozygous. But in this case, I'm seeing dominant. So it's brown, or black. And we refer to that for the dominant by uppercase. Like this is homozygous. Both of them are the same. Both of them are capital letters or uppercase letters. Homozygous recessive, one by one. Homozygous means similar or same, but recessive. So both of them are the same, but recessive, we refer to the recessive by lowercase. Hetero means different, hetero. So this is dominant and this is recessive. So it can be any of those. Here is the genotype and phenotype that I told you. Core of that chapter you have to note. This is the genotype. If I ask you, what's your eye color? Brown. That's phenotype. I look at your eyes and I say, this is brown, this is blue, this is black. Yes, this is observable traits or functions. Observable traits, whether it's physical or functional. This is observable. Do you mean observable? Observable like we can see it with our own eyes? Not necessarily. You can observe it with your eyes. You can observe it under the microscope, but there is a way to observe it. Otherwise, if you're talking specifically about the gene, the composition of the gene, this is called genotype. So if I tell you my eyes are blue, what's my phenotype? You just said your eyes are blue. This is phenotype. If I ask you what's my genotype, you say it's BB, lowercase, recessive, homozygous recessive. And again, observable function or physical trait, it doesn't have to be something that you see with your eyes, like blood type. Can you see the blood type with your eyes? No, but this is phenotype because it's expressed already. As long as you're not talking about the gene composition, this is observable. Disease, if somebody's having Down syndrome, disease, this is phenotype. This is something observable. If you study his genes to see what in the genes is responsible for that, this is called genotype. So here it is what we just talked about, location of a specific gene in a specific chromosome, alleles, which is a pair. This is the chemical form of the gene. At correspondent loci, same locus. Can be homozygous or heterozygous. Homozygous, both alleles, same thing. Uppercase B, or lowercase b. Hetero, different. One uppercase, one lowercase. Dominant is the one that's going to mask the recessive, like brown mask the blue. Genotype is what the alleles itself on the chromosomes that you can see. Phenotype is what is expressed already and it is observable. In order to uh, do this prediction that I just talked about, there are different ways to tell. Like if I give you like a phenotype, not a genotype, a phenotype, and uh, and I want you to predict. Like here is here is the dad, here is the mom. Can you predict what are the chances of the kids? Are they going to be like this or like that? Yes. we can do it in a Punnett square. We can know the outcome, but remember this, we can know possible outcome, possible. And there is another way, but this is only for experiments. It's not the most important. We are going to understand a little bit about it, but it's not the most important because this is more for deeper experiments. You're going to do experiments regarding, let's say, plants or flowers or something like that. The other way is the pedigree chart. Researchers use that like like the pedigree, like the family tree, for example. Here are the grandparents. This grandparent doesn't have the disease, and this parent have the disease, and they together they give three kids. So what are the chances of each one of those? And those one of the kids is coupled with another person, and they are going to produce another generation. What are the chances? So you follow two or three generations, and you can tell the possibilities. Probability, all these terms are important terms to understand. You have to know it. Probability is to measure the chance that a particular outcome could occur. So we're measuring the chance. Like, what's the probability of this person having Down syndrome? 25% of the outcome. So you measure the chance. Do you know 100%? No. That's why we call it probability. Most likely, the chance, measure the chance. And it's always ratio. So for monohybrids and bihybrid or polyhybrid, not the most important thing, but let me just give you an example here. Let's say you are matching those two together, these plants together, vegetables, and you wanted to know, or actually you wanted to uh, guess or measure the probability. So what's going to happen here is, let's say this yellow, and we know that these yellow beads are the dominant, and this green is recessive. So if one dominant, one recessive, if you do the Punnett square, can you tell me the chances? Yeah, we can tell the first generation. Like, this is yellow, and we know that it's homozygous dominant, uppercase Y, which is this one here. And the other one, which is green, lowercase. We know that. So what are the chances? This with this, uppercase, lowercase. Remember that this is dominant, yellow, so it will be yellow because the dominant masks the recessive. How about this one? Y dominant, Y recessive, of course, yellow. Dominant, Y recessive, yellow. Dominant, recessive. So we call that 100%. But this is monohybrid. What's monohybrid? One difference. Hybrid mean two different things. Two different things or two different variants. In what? In this case, it's just the color of that bead or that vegetable. That's it. So it's mono. While if it is more than that, you can predict two different things, and this is going to be the dihybrid. We will see it. But for monohybrid, for let's say this is cleft chin, like this. Some people have the cleft chin right here, and some others don't have it. Cleft chin, is that dominant or recessive? It's dominant. What do you mean by dominant? I mean, if I have dominant and dominant, you will get it. Dominant and recessive, that is heterozygous, you will get it. Recessive, you will not get it. But in this case, and we're talking about one thing only, so it's monohybrid. So here it is. Let's say this is one of the parents who is homozygous recessive. What's homozygous? Both of them are the same, not different. What's recessive? Lowercase. The one that's weak, that can be masked. So here is, let's do the chances. Here is one chromosome. You will get either this one chromosome or the other chromosome. You can get one of those, but both of them are the same, so it's not really different. And here's the dominant one who have cleft chin, uppercase, uppercase, both of them. It's homozygous dominant. 100% will be heterozygote, and 100% will inherit cleft chin, cleft chin. So the monohybrid, you can simply say, if it is like in these cases, it's homozygous with homozygous or this, it's 100%. If it is heterozygous, it's not 100%. It is 3-to-1 ratio, like it's 25%. Like, look at this. This one here, this is heterozygous, uppercase, lowercase, uppercase, lowercase. So what are the chances? Uppercase can come with the uppercase. Remember, this is heterozygous, and this is also heterozygous. So in homozygous, it's 100%. In heterozygous, it is 25 to 75%. So if the dominant come with the dominant, okay, you got it. If the dominant come with the recessive, you still get it because there is one dominant. So this is homozygous dominant. This is heterozygous, and you will get it. This with this will be heterozygous. This green here, heterozygous, you will get it. And the only chance that you don't get it is if this one goes with this one. Both of them are recessive, so this is homozygous recessive, so it's 3-to-1. So what's actually going to happen? You can be this, you can be this, you can be this, you can be this, you can be this. You can be any one of those four. So you have 25% chance to be this one, 25% this, 25% this, 25% this, or 75% you will have it, 25% you don't have it. You need to understand the following. So what are the chances? 75% you have it. What are the chances that I don't have it? 25%, which is 3-to-1. But listen to this. The chances does not change with more kids. Meaning, I have the first kid that have this cleft chin, and my second kid have cleft chin, and my third kid have cleft chin. So now I know that the fourth one is going to be non-cleft. Absolutely not. No, this is not how it works. Each one of the kids have the same chance. It doesn't change because it's random. Remember the random assortment that we talked about and the crossover, the two things that make us different. There are chances, and the chance doesn't change. Translation. What are the chances of my first kid to have the cleft chin? 75%. What are the chances he doesn't have it? 25. How about my second kid? No difference. Same chance, and so on. Here is if one of them is heterozygous, as you see here. So you will end up having 3-to-1. One of them only, this one is homozygous recessive, and only if that the case, you will not have it. Look, he doesn't have cleft chin, but those three will have it. 75 to 25 or 3-to-1. Here is what I just mentioned. Most probable outcome doesn't have to occur. So if it is 75%, can I have all my 4 kids without? Yes. You never know. It just probabilities, chances. It doesn't change. Dihybrid is, we are talking about two different genes at the same time, two different alleles. It's a combination of different things. So let's say we're talking about the dimple and the cleft chin at the same time, both of them. So this is called dihybrid. And you don't have to like know all these details, but it is just the chances. Like you have 9 out of 16. Uh, Instead of having 2 versus 2, it's 4. It's a double because we're talking about uh, two different genes, not just one. So it's going to be double. So it's 4 versus 4. If you count 1, 2, 3, 4 by 4, that's 16. Only one of 16 will not have anything. And then 3 smooth with dimple, 3 cleft with no dimple, 9, which is the major part, uh, uh, will have cleft chin and dimple, both of them, because both of those are dominant. This is a multi-hybrid. I'm not going to go through it. This is too much for this course. Just understand that we can talk about 3 genes at the same time. Doesn't have to be one or two. It can be more. So just understand the idea. Multi-hybrid, you're matching two different traits, but you're matching more than one gene at the same time. Like we're matching the cleft chin, the dimple, the ear. Is it attached at the bottom here or not? And something else maybe you can get more different things at the same time. This is called multi-hybrid. So if you know the genotype of the parents, you can guess or you can do the probability of the kids having these traits, which is how the phenotype will look like. Can anybody guess what is this here from the results? If you see the results, can you tell the parents? This is male Steve and female Wanda. If you know already that the male is having he's homozygous recessive. So both of them are recessive. And this one having one R. What is this? What do you think? Is it the same like this? Or is it lowercase? Does anybody know? Are you guys following? Does anybody know? You don't know?
It'd be lowercase.
It's lowercase, yes. It has to be lowercase. Why? Because of this. This is lowercase, and this has to be lowercase because you have this one right here. And if that's the case, what's going to be this one? Now this is lowercase. What is this?
2 lowercases.
Yes, two lower cases, exactly. The pedigree is something like this, and I want you to understand the basics. And the basics here are when you go like horizontal like this is horizontal, this is horizontal. So you're talking about generation. The pedigree is done in actual people with mating pairs, male and female together. Like this is the mom and this is the dad in the same line. So this is horizontal. Here is female, female, male, male. In the same line, we call that horizontal. Circle is how we represent females. Square is how we represent males. Moving vertically, moving down, like this is the grandparent generation, this is the parent generation. If you have a third one, this will be the kids. So moving from generation to generation to generation, we call that the vertical branching. Um, Again, circle is female, square is male. If you see it empty, completely empty, it means it doesn't have the trait. If it is black, it have it, or she has it, or whatever. If it is 50-50, it means one of this and one of that. This is how we do this demonstration, okay? Like this example here, this with this will give you these, and this is one generation, second generation, third generation. I'm not going to go into more details than that. I just want you to understand the basics of the pedigree and how can we predict. So if the black come with the black, we'll give you black. The white with the white will give you white, and so on. And like this will be female, and this is male. Good enough for this one. So for the punnet or pedigree, which one that we make it in the form of squares? That's the punnet. Pedigree is the generations that we talked about, horizontal and then vertical, circle, circle, which is female, versus square, which is a male, half and half, or white or black. This is, of course, the Punnett square. Monohybrid and dihybrid, are you talking about one or two genes at the same time? And this is basically you're talking about genotype, because we are talking about we are studying the genes for this, like XX, XY, B uppercase, B lower. We're talking about the genes. So and this is used for testing subjects. Um, what is the measure of the chance that a particular outcome could occur? It can be expressed as ratios or percentages. He's giving you a lot of clues, like 25 to 75, 1 to 3, and so on. This is the probability. It's the measurement of the chances. Measure of the chance that a particular outcome occur or does not occur. Most people outcome does or does not have to occur. Have or doesn't have, because we don't know. Sometimes it happens, sometimes it doesn't happen. Which one is a chart that's used by genetic researchers to trace trait through generations? Generations is obviously the pedigree. Okay, so far, we talked about one type only, which is one dominant, one recessive, dominant, mask, the recessive. Is that always the case? No. It doesn't have to be one dominant and one recessive, and that's it. No. In many ways, in many types, a lot of times, most of the time, it is not that simple. What we just talked about is the simple dominant and recessive, and we'll call that complete dominance. Brown hair, that's dominant. Brown eye, dominant. Blue eye, that's brown. They come together, dominant, completely take over, completely mask. So this is uh, dominance, complete dominance. It's if it is incomplete, you would say like mix. If it doesn't mix, here is one dominant, one recessive. If they come together, that's it. One of them prevail and the other one disappear. Doesn't have it will have zero effect. Zero effect. The dominant completely take over and kick the other one, completely mask it. But this is not always the case, because sometimes you will have incomplete dominance. Like, yes, this one is dominant and this one is recessive. When they come together, it's not going to mask it completely. There is a chance that doesn't mask it, and you get a mix of those. And sometimes co-dominance, like this is dominant and this is also dominant. Well, what's the issue now? If both of them are dominant but different, you can end up having both of them at the same time. And I will show you. And there is also something called pleiotropy that you have to understand. You need to understand these terms. All what I'm talking about is important. Core of the chapter. What is pleiotropy? One gene affects several traits. It's not like this gene for this trait only. This gene is for the blue eye. Yes, this is not pleiotropy. But this gene can make this person having three different issues. He will have the spleen that's bad. He have weak muscles. He will have small eye, for example. All three at the same time. Yes, this is called pleiotropy. Pleiotropy. It's not one gene for one characteristic or one trait. No, one gene, multiple traits. That's more valid in diseases or syndromes. You will see it. Versus incomplete dominance and codominance, I will show you, which is not the same dominance, which is called complete dominance that we saw. One more time, if it's complete dominance, when you have heterozygous, one dominant, one recessive, dominant, mask, the recessive, 100%. This is 100, this is 0. But sometimes it's not the case. It's not complete dominance, it's incomplete. So both of them will show up in different percentages. Or both of them will show at the same time, and I will show you what I mean. First of all, this is pleiotropy. One gene responsible for more than one trait. Marfan syndrome, if you ever heard of it. Marfan syndrome. And in this Marfan syndrome, this person will have like in one gene have the issue. A gene called FBN1. You don't have to know the details. Just understand one gene in one chromosome. In this case, a bad gene in chromosome number 15. Just one gene only. But he will cause many traits, issues, poor eyesight, weak aorta, long arm, long hands, long feet. All of this together from one gene only, we call that pleo. And pleo, by the way, means multiple. Pleiotropy, multiple traits. One gene, multiple traits. There is also the multiple allele system, and that will explain the codominance. Like if you look at your blood type, you can be A, your blood type, if you did your blood type, can be A, can be B, AB, or O. We all know that. There's one of the four. But this is a phenotype because this is what's observable. And you don't look at the chromosomes at this level, or you don't look at the genes at this level. You're looking at an outcome. This is called phenotype. Now let's take the chromosomes and look at it, which is the genotype. If you are A, I have two alleles, and both of them are dominant. Oh, this is not like one dominant and one recessive. No, both of them are dominant, and both of them can show up. Both of them are dominant. Both of them can show up, unlike the complete dominance. The dominant allele, 100%. The recessive is 0. Okay, this is one type, complete dominance. This is called co-dominance. A is dominant, B is dominant. Both of them are dominant. So if both of them come to one person, like myself, I'm AB. What's my genotype? It has to be AB. This is the only chance because both of them are showing up. So A is not like masking the B. No, because B is dominant. How about B masking the A? No, B is dominant as well. So this is called co-dominance. Both are dominant and everyone will show his own characteristic. How about AO? A. How about AA? A. Why? Like AA, both of them are A, definitely or A. What about AO? O is the only one that's recessive. So if you got one dominant, one recessive, yes, you get it. Or two dominant. But A and B, both of them are dominant, co-dominance, multiple allele system. You have three different alleles, not just one. You have A, you have B, you have O. And here A and B are dominant. So if you're B, you have two chances. BO, B is dominant, and O is recessive, you get B. or both of them are B. That's okay. The only way to get the O type is if you have both of them O. So incomplete dominance and codominance. Codominance is what I just talked about. And I will give you another example. But what is incomplete dominance? Incomplete dominance is this one is dominant, this one is recessive. The way we talked about it before, complete dominance. This one will show up 100%, and this one is 0. This is complete dominance. Brown eye, blue eye, 100% brown eye. Because this is too weak and this is too strong. Complete dominance. One of them is completely dominant. But incomplete dominance means this is dominant and this is recessive, but the recessive here is kind of strong a little bit. So you cannot kick him off completely. You cannot mask him completely. He will show up to a certain degree. Like, look at this flower here, or this rose. This is pink. Guess what? The parents, one of them was white and the other one is red. So what is this? Pink. So the white showed up and the pink showed up. Yes, but they are merged together. Merged to give you something in between. Look at this lady here. Wavy hair. Guess what? There is one of the parents have straight hair and the other one have very curly hair, and they mixed together. None of them is completely dominant to the other. One of them is kind of, like both of them will show up somehow. So you got this wavy, which is not straight and not very curly. Codominance is different. It's not going to mix and they show like part of this and part of that. No. Codominance, both of them will show up at the same time. No merge. So in this case, codominance, let's say the blood types that we talked about, for example. One is A and one is B. What are you going to get? A or B? Both of them, because both of them are dominant. This is called codominance. Another example, look at these cows. White, brown, it gives you white and brown. It didn't give you beige colored. If this is beige colored, this would have been incomplete dominance. Let's say the brown is dominant and the white is recessive, and then you get something in between. This is incomplete dominance. But here, 100% of this is brown. Some areas are 100% brown and some areas are 100% white. So both of them show at the same time, not a mix. So I hope you guys got the 3 scenarios plus the pleiotropy. Complete dominance, like uh, brown eye versus the blue eye. Incomplete dominance, like the flower. White, red, give you pink. Codominance, brown, white, you have patches of this and patches of that. Or blood types, A, B, both of them show up. So none of them is dominant and the other one is recessive. No, both of them can be dominant and both of them will show up. The other thing also that can happen is something called, again, it's getting complicated because it's not as simple as dominant, recessive, dominant, masculine, recessive. That's it. No, it's more complicated than that because there is something called penetrance. Penetrance. The penetrance is the degree to which the gene is expressed. The gene can be not expressed at all, can be 50-50, can be 100%, it can be 75%, it can penetrate to the next generation or not. This is another thing. Meaning that the genes can actually interact and they can represent themselves, or one of them can disappear, one of them can be 0 and the other one can be 100, because they penetrate from generation to generation. So the gene vary due to the gene interaction. And there can be also non-genetic factors. It's not completely genes, okay? I will give you guys examples of that. Like the environment can also play a role. Okay, like if you're talking about a parent who's tall and another parent who's short, is that the only thing that determine? No. How about the environment? What if this person is not eating enough to get, like, he actually got the gene to be tall, but he's not tall? What do you mean? He got the gene to be tall, but he's not? Yes, the environment. He's not eating enough to grow and become tall. This is example here. So in this case, it didn't penetrate, or it didn't show up completely. Can be percentage. So penetrance is uh, the probability of people who inherit an allele, what it express the traits or not. It can express, it cannot. Like here is cystic fibrosis, if you ever heard of this. In this case, this disease is known as 100% penetrance, unfortunately, meaning parents are homozygous recessive. This is lowercase c, and the dad is lowercase c. The parents will have the kids will have cystic fibrosis. Their kids will have cystic fibrosis. Their kids will have anybody who's having homozygous recessive. If you got the lowercase c and the lowercase c, that's it. But in another example, like polydactyly, poly means several, dactyly means finger. So this is the extra digit, a finger or toe, or a digit, I mean. So some people will have an extra digit, like here is an extra digit right here, extra finger, or an extra digit in the lower limb. They have 6 instead of 5. I'll show you the picture. But this does not have complete penetrance. So, what do you mean by no complete penetrance? Like lowercase p, lowercase p. So both alleles have this polydactyly. It doesn't show up. Yes, it can happen. So this is called incomplete penetrance. Like this is homozygous dominant, heterozygous, and this is homozygous recessive. Does this mean that this one will have the disease? In case of cystic fibrosis, because it has complete penetrance, yes. But in the other example, not necessarily. Like this polydactyly. If we put the same thing here, like if you have the two lowercase f, you will have polydactyly, not necessarily. You can have both of them, and you don't have to have it because of the incomplete penetrance. And this is polydactyly is the best example for that. Is it just the genes? No, I just give you an example. Maybe you got a gene to be tall from your mom, and you got the gene to be tall from your dad, so 100% will be tall. No, there is also environment. Same thing for weight, intelligence, skin color, eye color. It's not 100% genes. It can be polygenic. Poly means multiple. So it can be multiple factors. Like these eyes, for example, uh, there can be um, other factors influencing this. Multifactorial can be also for environment. I give you example of height, for example. Both parents, you got allele for both from both parents to be tall, and you're not tall as expected. Environment, about the environment. Maybe you didn't get enough protein to be tall. It's not 100% genes. We call that polygen. Maybe you have injury that caused you to lose your growth hormone. But wait a minute, I have the two genes to be tall. Yes, but you don't have the growth hormone. There is another thing. Skin color. If you're exposed to tanning, your color is not going to be exactly as expected. It can be darker. Um, Diseases, for example, if you have the genes for the disease, are you going to get it or not? Maybe you don't have enough of the genes, but you are exposed to harmful things and you get the disease. You get cancer, for example. Twin studies, we're going to talk about the twins. I'll just give you a brief idea and then we will do the rest next time in part two. Twins. Remember, the normal is one sperm fertilize one egg, you got one zygote, which will become one fetus and one person. This is the vast majority. Sometimes that doesn't happen. Sometimes one sperm uh, fertilize uh, two eggs, or fertilize one egg that divide fast. Or sometimes, and this is called identical twins. This is identical twins. But we hear about the fraternal dizygotic twins. These are twins that they do not look like each other. This is identical monozygotic, and this is fraternal, which is dizygotic. So what happened here is there are a lot, millions of sperms. So it just happened that um, two eggs are produced at the same time. Can happen. So obviously, because there are millions of sperms, so if the female produce two eggs at the same time, a sperm will fertilize one, another sperm from all these sperms available around will fertilize another egg, and you end up pregnant with a twin that's not identical because it's two different eggs, two different sperms. But here it fertilized it and it grow and split fast and ended up giving you two. They will be identical, and that's good enough for this part. Let me stop here, and the rest will be done next time.